Good morning, everyone. Today, I'm going to take class about malaria. It comes under infectious disease, general pathology. We'll start with the lesson plan. First, we'll go for the epidemiology, followed by mode of transmission, names of the Plasmodium species, life cycle, its clinical features, how to diagnose it, treatment, and prevention part. Epidemiology. Malaria is endemic in tropical and subtropical developing countries. It is a major health problem. Every year, 300 to 500 million cases of malaria with 1 to 2 million deaths, most of them in Africa. Now, this is the world map. You can able to see the entire South America, Africa, and India, all those, even the Asian parts are involved by the malaria. Malaria, the mode of transmission of the malaria is mainly by the bite of female Anopheles mosquito. So the bite of the female Anopheles mosquito will lead to the malaria disease. And other rare modes other than bite, it includes blood transfusion, use of contaminated syringes or needles, even congenital from mother to fetus, malaria can be transmitted. So, the mode of transmission is mainly by the bite of the female mosquitoes. Other rare methods are blood transfusion, use of contaminated syringes or needles. Congenital is from the mother to fetus. Next, we'll go for the species of the malaria that infect the humans. There are 5 species are there. First, we'll see the Plasmodium vivax, which is the most common. Almost 70% of malaria it is caused by the Plasmodium vivax. Followed by Plasmodium falciparum. It includes 25 to 30%. Mixed of Plasmodium vivax and the falciparum, it includes 4 to 8%. Beyond the vivax and the falciparum, other species are Plasmodium falciparum, Plasmodium ovale, and Plasmodium knowlesi. These knowlesis are included only in the Southeast Asia. Next, we'll go for the life cycle of malarial parasites. There are 2 life cycles. One is asexual reproduction and other is sexual reproductions. Asexual reproduction occur in the human and sexual reproduction occur in the female Anopheles mosquitoes. Coming to the asexual reproduction is also called as a cytogony. It is a process of reproduction. Humans are the intermediate host of malarial parasites. Human cycle begins with the bite of female Anopheles mosquitoes. Once the female Anopheles mosquitoes bite the human, it injects sporozoites into the circulations. Once the sporozoites enter into the circulations, because of the bite of the female Anopheles mosquitoes, that human is considered as an infected human. And in the infected human, these sporozoites affect primarily in the liver cells, and also the red blood cells. In there, the sporozoites, once it enter into the liver cells and the red blood cells, it is called as a cytons, right? And let us see individually what is happening in the liver and what's happening in the red cells. In the liver, it is called, as I previously told, it is called as a hepatic cytons. This hepatic cytons undergoes binary fission and it produces the merozoites. These merozoites will be inside the cytons. Once it is more number of division of the cytons occurs, there is a more number of merozoites will accumulate inside the hepatic cytons. Later, there will be a burst in the merozoites. One hepatic cytons will releases almost 40,000 merozoites. So now this picture clearly tells as a bite of the female Anopheles mosquitoes, it injects sporozoites into the circulation. This sporozoites, once it enters into the human being, it is called as an infected human. The sporozoites will enter into the hepatic, excuse me, the sporozoites enter into the circulation, then it enters into the liver. In the liver, it is called as a hepatic cytons. Under binary fission, these hepatic cytons will undergoes binary fission and is converted into merozoites. Then this merozoites rupture of the merozoites will be occurring, and once the merozoites are ruptured, then it will enter into the red blood cells into the circulation. This is the one part of the cycle. The merozoites, once it enters into the red cells, it will utilize the red cell content for their metabolism and become trophozoites. The trophozoites, by multiple nuclear and the cytoplasmic divisions, the cytons, again it will be converted into cytons. This cytons mature and releases the merozoites, and once the released merozoites will enter into the red cells, and once it is released along with the merozoites, the red cell content is also out, malarial toxins is also out, and malarial pigments is also out of the mature erythrocytic cytons. So now this picture clearly tells once the hepatic cytons with the merozoites ruptures, the ruptured merozoites will enter the red cells, uses its content for the maturations and become trophozoites. Then this trophozoites, by its maturation, it become cytons, and you can able to see the cytons, and inside the cytons there is a merozoites are multiplying, and at the end the merozoites are released from the red cells, and again this released merozoites are entered into the circulation and is trying to affect the other red cells. Some merozoites, instead of developing into trophozoites and cytons, it may transfer to the male and the female gametocytes, which are the sexual forms of infective malaria mosquitoes, which are infective to the mosquitoes. The person who are harboring this are called as a carriers. Usually, these hematocytes are not pathogenic for humans. Now, this is the entire cycle, asexual reproductive cycle. As soon as the sporozoites enter into the circulation, into the hepatocytes, the sporozoites will multiply, and there will be a merozoites productions by the binary fissions. Again, there is a, once it mature, the cytons mature, the merozoites are released in the circulation, and these merozoites which are released in the circulation will enter the red cells, mature to the trophozoites, and mature to the erythrocytic cytons. These, once the erythrocytic cytons mature, it releases the merozoites into the circulations. These merozoites, not all the merozoites are burst. Some of the erythrocytic cytons will enter into the phase of gametocytes. That is, it will be mature to the male and the female gametocytes. The person with the male and the female gametocytes are called as carriers. Once the blood meals are taken by the mosquitoes, the gametocytes enter into the mosquito's stomach. So far, we have covered the asexual reproduction of the malarial parasite. Now we are moving on to the sexual cycle. Sexual cycle is otherwise called as a mosquito cycle or sporogony. Blood meal from the infected persons, male and the female gametocytes enter into the mosquito's stomach. One male gametocyte will produce 4 to 8 microgametes in the stomach of the mosquitoes by eggs flagellations, and female gametocyte will mature to produce 1 macrogram gamete. These male and the female gametocyte will be attracted towards each other by chemotaxis, and there is a formation of the zygote. And once the zygote is formed, it may mature to form ookinete. These ookinete will penetrate the stomach lining, comes outwards to the stomach, and it produces an oocyst. And further growth and maturation of the oocyst, multiple sporozoites are formed. After complete maturation, oocyst ruptures and the sporozoites will be entering into the circulations. Most of the sporozoites will migrate to the salivary glands, and it will be there in the saliva of the mosquitoes. And the infection will be transmitted to the human by the bite of the mosquito. Once the mosquito bites, along with the proboscis, some saliva also will be entered into the circulations. So the sporozoites which are present in the saliva is also will be transmitted into the human. This is the sexual reproduction. This picture clearly tells about that. So this is the by the bite of the mosquito takes the blood meal. So once the blood meal is taken, the person with the gametocytes, male and the female gametocytes, united by the chemotaxis, it produces the gamete, and this gamete will mature to the ookinete, and once it penetrates outside the stomach, it produces a oocyst. In the oocyst, it mature into the sporozoites and enter into the circulation. Most commonly, it enters into the salivary glands, and once the mosquito bites, this sporozoites which are present in the salivary gland will be entered into the human body. So the same picture, it can clearly tells it's a sexual reproductions, bite of the female Anopheles mosquitoes. Gametocytes are entered into the stomach, and in the stomach, it is penetrate in the stomach, it may become ookinete. When it penetrate outside the stomach lining, it is called as a oocyst. And then the oocyst will be oocyst develop into the sporozoites, and the sporozoites will be entered into the circulations, and these sporozoites will be most often, it will be seen in the salivary gland. So we are ending with the life cycle of the malarial parasite. Life cycle of the malarial parasite, it has sexual and asexual reproduction. Asexual reproduction occurs in the human. Humans are the intermediate host. Sexual reproduction occurs in the mosquitoes. In the asexual reproduction, the human starts with the bite of the female Anopheles mosquitoes. Sporozoites are inoculated into the human's body, and from there it enters into the hepatocytes. There it is called as a hepatic cytons. It may mature and releases the merozoites. These merozoites enters into the red cells, and it uses the content of the red cells and become erythrocytic cytons, and again their merozoites are produced. These merozoites are ruptured, and the cycle will progress. Again, the uh, will affect the erythrocytes, but some of the erythrocytic cytons will remain as a gamete and may develop into the gamete, male and the female gametes. The person with the male and the female gametes are called as a carriers. When a bite of the female Anopheles mosquitoes, these male and the female gametes will enter into the stomach, and in the stomach it may united to form a zygote with the help of chemotaxis, and the zygote which is formed end up with the ookinete, and this ookinete will perforate the stomach wall, and there it is called as a ookinete oocyst, and these oocysts will mature to the sporozoites, and the sporozoites will be released into the circulation of the mosquitoes, and these are more transported to the salivary gland of the mosquitoes. The bite of the female Anopheles mosquitoes along with the proboscis, saliva will be entered, and along with the saliva, the sporozoites also will enter into the circulation. This is the life cycle of malarial parasites. So hope so I have done elaborately a teaching of a life cycle. Next, we'll move on to the symptoms of malaria. They complaints of fever with chills, sweating, and headache, dry cough are some of the symptoms of malaria. This malaria has a paroxysms of high-grade fever along with anemia and splenomegaly. They have three stages. One is cold stage, and it lasts for 20 minutes to 1 hour. It's a sudden onset of fever with shivering, and followed by a hot stage, which includes 1 to 4 hours. The fever, it is a fever stage, it may reaches the maximum. This fever is always associated with severe headache and the body ache. Sweating stage, it is 2 to 3 hours. The patient will be complaints of profuse sweating and the temperature falls. So malaria has three stages. One is cold stage, followed by hot stage, followed by the sweat stage. Cold stage, which lasts for 20 minutes to 1 hour. So sudden onset of fever will be there, and hot stage, which includes 1 to 4 hours. The maximum fever is recorded, and it is always associated with a severe headache and body ache. And followed by sweating stage, it will be the patient complains of profuse sweating and temperature falls there. So far we have done with the clinical features. Now we are going for the complication of falciparum malaria. Complications are, if it is involvement of the cerebrum, then it is called as a cerebral malaria. The cerebral malaria, the patient will be complaints of hyperpyrexia, that is a high grade fever, then followed by convulsions, coma, even sometimes the patient can go for death. If there is a sudden onset of massive intravascular hemolysis, then because of the intravascular hemolysis, there will be a hemoglobinemia, hemoglobinuria, hyperbilirubinemia, and high-grade fever. So finally end up with the black water fever. And if it is a renal involvement, can have a renal failure. The plasmodium malaria, it can lead to the nephrotic syndrome. So these are the complications of the malaria. So complications of the malaria, it includes cerebral malaria, black water fever, renal failure, plasmodium malariae. So far, we have done with the clinical features, complication. Now we are going for the methods of diagnosing malarial parasites. We'll start with the medical history. If the patient is having 3 stages of the fever and the patient is from the endemic place, then you can go for the diagnosis of the malaria, and followed by physical examination. If anemia, splenomegaly, then they can go for the diagnosis of the malaria. So that is the medical part. Now in pathology, a microscopic examination of the blood smear is the diagnostic part. And along with the microscopic examinations, there are other methods also, that is rapid diagnostic test. Rapid diagnostic test, it is mainly with the help of commercially available kit. And detection of the nucleic acid of malarial parasites by the PCR reactions also identified. Even fluorescent microscopy can be used for the detection of the malarial parasites. We will see in detail about the microscopic examination of the malarial parasites. Microscopic examination of the malarial parasite, it is the gold standard for the diagnosis of the malaria. Ideally, for examining this, the blood sample should be collected from the skin punctures and smears are made immediately. If there is a delay in the making smears, there may be morphological changes in the parasite, so it will be difficult in diagnosing. 2 types of smears are made for the detection of the malarial parasite. One is thick smear and other is thin smear. Thick smear, with the help of thick smears, we can able to detect the presence of malarial parasite. With the help of thin smears, we can identify the species of the malarial parasite. So, microscopic examination of the blood smear for the malarial parasite is important, gold standard for the diagnosis of the malaria. And 2 types of smears are prepared, thick and the thin smears. Thick smear is for the detection and thin smear is for the identification of the species. So this is the picture. You can able to see a doctor is trying to make the smear with the skin puncture, and he will be doing the smear immediately in order to prevent the morphological changes of the malarial parasite. This is the thick smears. With the help of thick smears, we can able to see the presence of the malarial parasite. Thin smear, this is the below, it is a thin smears, and the thin smear is used for the confirmation of the species of the malarial parasite. And these parasites, bivax, will have affinity for the N blood cells. Malarial and oval will be having bivax. Oval will have the affinity for the old red cells, and falciparum, it affect all stages of the cells, even it affect the young red cells and also old red cells, and it is more dangerous compared to the falciparum, malaria and oval. So once a smear is made and it is stained with a Leishman stain, then how to recognize the malarial parasite in the smear? So inside the RBCs, you can able to see a red chromatin dot and the blue cytoplasm. So this is how we can able to identify the malarial parasite. Along with the, there can able to see various stages of the Plasmodium species inside the erythrocytes. We can see a ring form, or we can see the cygones, trophozoites, and gametocytes forms are seen. Cygones, again, your ring, you can see the like ring, we can see the chromatin dot and surrounded by the blue cytoplasm, and in the cygones, we can able to cygones and the gametocytes, we can able to see the malarial pigments. These malarial pigments are nothing but which is the brown black granular pigments. These pigments are nothing but it is the breakdown products of the hemoglobins. This can be called as a hemazoin pigment. Now you can see the RBCs. Here there is a hemazoin pigments which is sitting along with the cygones. These are the different stages of the malarial parasites found in the peripheral blood. So we can able to see the ring forms, cygones, gametocytes. So these are the different stages. The morphology of the ring forms in the falciparum, bivax, and malaria will be different. So that was a schematic diagram. This is the Leishman strained inside the RBCs. You can able to see the stages like this. Gametocytes of the falciparum will be a banana shaped. And here you can able to see the malarial pigments, brown black malarial pigments in the cygones and the trophozoites. So here also we can able to see the central chromatin dot and surrounded by the blue cytoplasm. Here multiple, this is might be a falciparum, because in the falciparum there will be a multiple ring forms and aqualine forms can be seen. So far we have seen the microscopic examination of the malarial parasites. To call the smear negative for the malarial parasite, it is recommended to examine at least in 100 oil immersion in case of thick smears, 200 oil immersion in case of thin smears. So we have to examine 200 field in an oil immersion in a thin smear to call it as a negative. In case of thick smear, we have to examine under oil emulsions, 100 field, we have to examine the thick smears. If we are not getting any parasite, then only you can tell the smear is negative for the malarial parasite. And there is a reporting format is there for the malarial parasites. First, we have to tell whether the blood smear is positive for the malarial parasite or not. Followed by which species is involved, whether it is a vivax, falciparum, malariae, which species is involved should be included. Then, in which stage of parasites, whether it is in the cyzones, whether propozoids, gametocyte, which stage should be included. And parasitic density should be told in the report. That is useful for the falciparum malariae. Other than microscopic examinations, other examination includes Buffy coat. Buffy coat, it is a quick and it is used for screening for the large number of the samples. And once the Buffy coat is made, when it is examined and stained with the fluorescent stain, you can able to see the green yellow color falciparum in the dark background. This is a malarial parasite stained with the fluorescent stain and seen under the fluorescent microscopy. There is a green yellow color parasite in the dark background. And next is antigen detections. These are commercially available, kits are available. So once you can place the serum or plasma, you can able to see with the control. The test is also positive, then you can go for the diagnosis of the malaria. But this made up, it includes either Plasmodium lactate dehydrogenase, and next is histidine-rich protein aldolase. So these three substances are used for the detection of the malarial parasite by the rapid kit method. We'll go for the treatment. So treatment for the malaria, we should have two concepts. One is falciparum chloroquine resistant and chloroquine sensitive. If the chloroquine sensitive, then this falciparum is treated with the chloroquine. If it is resistant to the chloroquine, then it is treated with the quinine, mefloquine, sulfoquine, and primaquine. Vivax, malariae, oval are treated with the chloroquine with the primaquine. So, finally, we are end up with the prevention of malaria. So first we have to prevent the mosquito bite by covering our limbs, using repellents, coils, sleep under the net, avoid stagnation of waters, and chemical prophylaxis can be given to the travelers to the endemic places. If it is chloroquine sensitive, then you can use the chloroquine as a drug. If it is chloroquine resistance, then you can go for mefloquine, doxy are used. So far, yes. So far, we have seen malaria, epidemiology. It is endemic in various countries, predominantly Africa, South America, India, and some part of Asia. There are 5 species. First is falciparum, vivax, oval, malaria, and knowledges. And next is we have dealt with the reproductions. And reproduction, it has 2 cycles. One is asexual and other is a sexual cycle. Asexual cycle that occurs in the human and sexual cycle that occurs in the mosquitoes. Then we have dealt with the clinical features like fever, chills, rigor, headache, body ache, and they have three stages. One is a cold stage, hot stage, and profuse sweatings. With this, we can have a diagnosis of the malaria, and this patient will have splenomegaly. And malaria can have a complications of cerebral malaria, black water fever, nephrotic syndromes, renal failures. And next, we have seen the various diagnostic modalities like microscopic examination of the peripheral smears, thick and thin smears. And next is a Buffy coat. Next, antigen detection by the commercially available kit and polymerase chain reactions, followed by the treatment for the chloroquine. Sensitive chloroquine is given. For the chloroquine resistant mefloquine is added. And next is and how to prevent it by covering our legs, use the mosquito repellents. And for the person who are traveling to the endemic countries also will be vaccinated with the prophylaxis of chloroquine, mefloquine, doxycycline are used. Hope you have understood malaria topic. Any doubts, kindly contact me. Thank you so much.